So screening assay development. So in order to select our assay or develop our assay, we need to consider a few factors. So the first factor, the first point we need to consider is detection of relevant target activity. So target activity means is depending on our target. For example, if our target was the enzyme, so we need to watch our enzyme activity. That means if the enzyme is involved in increasing ATP in the cell, that means we have if our ligand blocks the enzyme activity, that means the ATP will decrease inside the cell. So enzyme activity, are we want to detect that, or ligand binding, or if we want to see the only the binding of the ligand, not the activity, then we have to select a binding assay. Or transcription factor, if our target is a transcription factor, then we have to select another assay. For example, there are assay ChIP-seq, I guess, that accounts for binding of the transcription factor to the DNA. Another type of target is the dimerization. For example, if our target is the receptor protein, then there are receptors upon binding to the ligand, the receptor, one receptor, then another type of receptor, it can bind, two receptor can bind and dimerize. So if we want to block that dimerization of two receptors, then we can detect an assay that would account for dimerization or blocking of the dimerization. So the next point is the technology platform. Which technology are we using? For example, our assay is cell based. We want to use a whole cell or the part of a cell, that is a biochemical. For example, we can use a part of cell that is only containing our target enzyme, then that is biochemical assay, or cell based, the whole cell. So, and the detection technique, the signal technique, the assay result, we see the assay gives a light, that is it luminescence or fluorescence or colorimetric or radioactivity. Radioactivity is not that much used because it is dangerous. So luminescence and fluorescence, these two are quite used in assays as a detection of lights. Then we get signal that if a molecule or ligand binds to a target, then it emits light or fluorescence or luminescence. So depending on this, we have to consider. I have to also consider the manual or automated assay, because automated assays are much quite fast, and we can screen lot of compound. With automated assay, we can screen much more compounds at a faster time. Then also number of plates and readers is another fact. Depending on our assay, if our assay is capable of high throughput, then we can use 3000 up to 3000 well plates. If our assay is not that high throughput, then we have to select 96 well plate. And if our compound library contains less number of compounds, then we can select low throughput assays, which we can use 96 well plate, microtiter, or 384 well plates. So here is the assay throughput range. If our library is small, then we can use low throughput assays or mid throughput assays. But if our library is huge, like for example, if our library is containing millions of compound, then we have to select high throughput assays or ultra high throughput. So these two are mainly found in industries, drug companies basically. So yeah, there are a few questions we have to consider. So going to the next slide. So in the earlier slide, we talked about the cell biochemical assay, in vitro assays, but we can also use an in vivo model to screen our drugs. For example, this figure is a zebrafish figure, which is a transparent system, living body. So because of its transparency, we can visualize directly the effect of drug or effect of compounds on its bone density or blood vessel growth and other systems because of its transparent body. So the whole in vivo system can be used as a screening technique or screening assay. So obviously, in vivo are more clinically relevant than in vitro bioassays. So in vivo is required, because if zebrafish is not suitable, then we can always use a mouse model that will recapitulate our disease. So after testing testing the bioassays in vitro, then in the next step is to test them in vivo models, mouse models. Going to the next slide. So here is the basis of a screening assay. So a screening assays report some aspect of target activity. It could be binding or target or any functional change in a multi-well format. So this is microtiter plate, and we add compounds on each well plate and see the activity on the, it could be a light fluorescence light that gives a signal. So compounds added to plate 1 per well, and compounds resulting in a change in assay. So the compounds that heat that binds to our targets gives a readout. That means it emits light. So those compounds are heats. Then heats are then retested in secondary assays to further confirmation of activity or selectivity. Then we optimize those heats. Their chemical properties we optimize to for maximal activity that ultimately generate lead compounds. So moving to the next slide. So heat compounds are not that much good always. They have low affinity against our target with poor cell penetration, and they may have low solubility. That's why we need to optimize those leads, heat compounds. So these are further polished in the lead optimization stage. There the medicinal chemistry application being applied there, take different techniques, and assays mainly test the compound's binding affinity or potency. We can also extrapolate potency with those assays, and also we can see efficacy. So, so the type of assay mainly we use biophysical assays or cell-based assays or biochemical assays. We talked about biochemical and cell-based assays earlier, but in the next slides we will be learning these three types of assays in details. So choosing those different types of assays, that forms ultimately the screening cascade. That means one assay, then after assay, then what will be third assay, or the primary assay and the secondary assay. So this forms the screening cascade, and after testing through this cascade, that generates heat to lead, and the later stage is lead optimization. Then we talked about that at first we test all the compounds at a single concentration, then we get hits, and after setting the minimum criteria, then we select hits, and then we test them at different concentrations. So at testing them and in different concentration, we can find their dose response curve. I mentioned dose response curve, and from that dose response curve we can find their potency. That's IC50 inhibitory concentration, where 50% target is inhibited, or AC50 where the 50% target is shown effective activity. So this is quite important. In this figure you can see that in the x-axis there are a lot of concentration, range of concentration we used, starting from very low nanomole, and then the concentration is increasing, and in the y-axis there are cells. For example, in cancer context we see the cell line growth. For example, this is the survival cell survival. So at the very low concentration of the compounds, the survival is 100%. So no cells, so all the cells are growing, so no cells are dying. So at very much low concentration, the drug is not showing, the compound lead hit compounds are not showing their effect. So there is no effect. But as we increase our concentration, the surviving cells decrease. That means growth is slowing. So at this very high concentration, all the cells are dying. So there is no survival. So that is max effect. So at which concentration, if the 50% cells are inhibited or died, or 50% survived, that 50% concentration is called IC50. So each and every heats will have a IC50 concentration. So those heats that having IC50 concentration low, then those are highly potent. So that's how we generate dose response curve, and we extrapolate IC50 value of each of those heats. So and that's how we can get the potency of those compound in vitro. So in the next slide or next lecture, we'll be learning the different types of assays. For example, biophysical assays, which are NMR assay can be used, or SPR, surface plasmon resonance. Biochemical assays, different types of biochemical assays we use to screen the hits. We also use cellular assays or cell-based assays. So we'll be learning those in details.